right. Today I'll be presenting on behalf of the BioPilot group. I'm Mike. Other members are Josh and Charles. BioPilot is a co-pilot tool for end-to-end generative design of synthetic biology circuits. We've built this into the existing Knox code base, and I'll go through the overall technical view and a short demo here. So first, the general architecture is that the user supplies a plain English prompt. The AI agent loop then is hooked up to a parts layer, a design space engine, and a foundation model layer. The agent will then use these layers to pick parts, build designs, and potentially fill in de novo parts using the Evo2 foundation model. What this will ultimately produce is a browsable design space with buildable DNAs for each design, and we can generate alternatives for each part slot. Importantly, the agent never actually touches DNA directly. It only calls parts from a database and does, GOLDBAR based design and sequence replacement using the tools that we've created. So we're not subject to hallucinations, which would be a main concern for this type of application where it just makes up parts and says that they'll work. So, how this would work is the user would say in chat, design an arsenic biosensor in E. coli that produces GFP. That goes into the LLM, which reads the intent. The intent will, include, I need to get, like, the needs will include a need for parts. So it'll query the database to get parts, which are relevant to the request at hand. It'll then generate a spec or a design space GOLDBAR combinatorial spec and import it as a version graph. And from this, we can enumerate all the compatible designs in Knox. And then these enumerated designs are our buildable DNA. We render these as color-coded slots and also at this point can hook in our Evo2 model. Before I move on to that, basically this enables just sort of a plain language generation of a synthetic biology circuit. This saves the user lots of work in terms of you wouldn't have to go scrape together parts and, do the nitty-gritty of like, okay, I need a RBS for this, I need a primer for this, da 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 da. You just give a simple request and you'll get a circuit back, which is at least a lot faster to get to the starting line if you're going to then, optimize or actually assemble these circuits. Um, so our second capability is Evo2. You can imagine that we take one of the designs that we made using our first capability, and then, say we have a promoter, an RBS, a CDS, and a terminator, and I want to see if I can de novo create a new RBS that can, have a stronger, that can drive stronger expression of GFP. So I could do this by just clicking view in the Knox enumerated sequences. I could then click regenerate using Evo2. We'll call Evo2 from the NVIDIA hosted version of the model, and Evo2 will return a new DNA sequence with a mean sampled probability. And we'll have both the original and the Evo2 generation side by side, and the user can decide to keep, reject, or recreate. So Right. So originally we demonstrated option 5, I think. So in this case, I think I'll go ahead and demonstrate option 2. Let's design an arsenic sensing NOT gate that produces RFP. So I'll go over to Knox now. Design an arsenic sensing NOT gate which produces RFP. All right. So we'll give that a few seconds to work. Usually takes around, 30 to 45 seconds. I might speed up this part of the video. All right, we have our result. Let's see. Okay, we have a constitutive promoter, RBS, arsenic regulator, arsenic sensor promoter, lac I. Okay, RFP reporter. Okay, so how does this work? It constitutively expresses R as R. TU2 senses arsenic. R represses the arsenic responsive promoter. In the presence of arsenic, it's inactivated, allowing parts to express lac I. Lac I represses the PLAC promoter, and then lac I is expressed due to arsenic, or so when lac I is expressed due to arsenic, PLAC is repressed, which turns RFP off. So arsenic present, RFP off. This is a NOT gate. Perfect. Cool. And so we can check this out here. Okay, so if I go here, we can get arsenic sensing NOT gate, RFP, and all right, let me enumerate these designs. Enumerate. Okay. Okay, we only have 3 designs here. That's all right. Are they different? Oh, yes. Okay. Okay, yeah, we have a few different promoters to start. Okay. So here's our whole sequence. I'm just going to view this first one, and you can see we have all of our CDSs and our transcriptional units. So just for example, let's say I want to regenerate this part with Evo2. I can click regenerate, and here's the proposal. It's got a low probability. Here, what if I try again? Yeah, the same one. All right, so let's apply this proposal. Cool. We've edited it, and then I'll export it. And then we have a file that we can import, to classic sort of molecular biology tooling like Benchling, and it'll have all of the annotations that we have in here carried over. And that's, a nice starting point for, assembly or further tweaking. And yeah, that's all. Thanks for watching.